Okay, well, let's go ahead and get started. There are people that are still logging in, but want to welcome everybody to the 16th annual Early Career Scientist Symposium. If anybody has been obviously following the news, this was rescheduled from last March, but we're able to all get together again, or get together virtually. So I'm glad that's worked out, and it's actually worked out in terms of the reach. We were able to increase the number of participants drastically from what our typical in-person early career symposium includes. So there's approximately 500 people signed up for this. So it's exciting that there's such broad interest in natural history collections. Before I hand things over to Dan Rabosky for the formal department welcome, I want to introduce myself. My name is Cody Thompson. The mammal collection manager and assistant research scientist in ecology and evolutionary biology at U of M. I'll be moderating today's session. The format of our sessions, format for the session today, as well as future sessions, which will occur every Friday now until April 2nd from 1 to 3, will include a series of presentations from our invited speakers, followed by Q&A and a panel discussion, which will be led by various members of the Early Career Committee. The panel discussion is meant to engage all the participants that are here. So if you'd like to ask questions of any of the panelists, please submit those questions to the Q&A box, and we'll get to those as they come. If you have specific questions that you want to direct towards a specific panelist, please add the @ sign and with the person's name. That way we know to direct that question to them. We'll address any questions as they come, and hopefully it generates some lively discussion between the panelists and the moderators, and hopefully help better inform the audience about natural history collections and why they're such an important aspect of academic biology. So without further ado, I'll turn things over to Dan for the formal introduction.
All right, thank you, Cody. So I am Dan Rabosky. I'm an associate professor and curator in the Department of Ecology and Evolutionary Biology, and a curator of herpetology in the Museum of Zoology. So thank you all for coming to this symposium. It's pretty awesome to have this lineup of speakers and topics that on this general theme of collections. And I know a lot of us have a lot of thoughts about the role of collections in the future of our field. So I'm going to just share my screen here and go through a few aspects of. Second delay here. So the theme of our symposium this year is natural history collections, drivers of innovation. So this is the 16th annual Early Career Scientist Symposium that we've had. Pretty excited about this one. It's a topic close to my heart. And so if you can go, I'm sure most of you have seen from the website the overall list of speakers and so on that we have, beginning of course today with Rob Gerolnick and ending on April 2nd. So I'd encourage you to peruse these titles and attend as many of these as you can. And just to give another recap what Cody mentioned in terms of format. So today with Rob Gerolnick's talk, we'll have the talk go from approximately 1:15 or 1:20 to 2 or a little thereafter. And then from around 2 to 2:45, we'll have questions and a moderated roundtable discussion with Rob Gerolnick and myself. And that general format will carry through for the duration of the symposium. The exact times, 2 to 3 for the roundtable discussion following the actual formal talks. I'd like to thank a number of folks for making this symposium possible and for their support, including especially the Department of Ecology and Evolutionary Biology for sponsoring the symposium. And I'd like to thank the other members of the Early Career Scientist Symposium Committee 2021. So that's Benjamin Nicholas, Teresa Pagan, Brad Ruffel, Cody Thompson, and Taylor West, as well as Linda Garcia, Gail Kuhnlin, and John Megahan, all of whom provided a number of aspects of logistical and other forms of support. And really, they were all these folks were essential in having this come together the way it has. I'd also like to call out some folks specifically from EEB for their contributions here and their help overall, especially our current EEB chair, Tricia Whitcop, and our chair through July of last year, Dermot Foyle, who was the chair when we had the original incarnation of this particular symposium theme come to fruition, although it was postponed. And likewise for our museums director, Hernan Lopez Fernandez, also played an important role in bringing this together. And he also served on the committee last year, so he essentially was a committee participant last year in the organization of the symposium, which is now happening, as well as Chris Stick, who is the museum's director through July of last year. So thanks to everyone for your help with helping to bring this together. And just to reiterate some of the basic format issues here. So we got a Q&A roundtable and the questions read by the moderator. Accessibility, you have closed captioning option. You should see the ability to get the live transcript down near the bottom of your screen there. And on Twitter, if you use Twitter, you have a hashtag here for ECSS, and you can tag EB if you'd like as well. So I thought I'd give a brief overview of Michigan Biodiversity Museums, just sort of a snapshot view of what Michigan museums are. So one of the reasons I think for those of us who are affiliated with the museum here, this is a tremendous place to work, and we have about 17 million specimens between the Museum of Zoology and the University of Michigan Herbarium. Many of our collections are among the largest university-affiliated collections worldwide. Some of the largest collections overall worldwide. This includes birds, insects, mammals, freshwater mollusks, all among the largest university collections worldwide. Fishes are the second largest collection in North America. We have a very large herbarium, and we receive a large number of visitors per year, over 5,000 research visitors per year, and hundreds of students training and studying and working in the collections on an annual basis. So a lot of activity. From a personal perspective, as a curator of herpetology, it's a tremendous privilege to work here because we are the second largest collection of reptiles and amphibians worldwide. So we have a spectacular research and teaching collection for reptile and amphibian biodiversity. And I'd like to add that one thing about working here that I think makes it really special is that we have a tremendous facility for work as well. So the university has invested very heavily in a new research center where our research collections are housed, along with some of the best working space that I've ever seen. For example, within herpetology and ichthyology, we have a fabulous range of basically a tremendous amount of space for working, and number of supporting resources and so on. And the university has really shown their commitment to the museums overall. This is just one example of the sorts of things that we've seen investment in recent years. This is a new CT scanner, dedicated CT machine associated with the Museum of Zoology that is housed in the wet divisions, so the ichthyology and herpetology collections. And so we're using this now to generate large amounts of high resolution 3D images for large chunks of the tree of life, especially vertebrates. But it's a general resource for the museum community. I thought, whether I wanted to give a very short little personal reflection on natural history collections and how it relates to some big questions of biological diversity, just to maybe echo a little theme that I don't know if will come up during the course of some of the talks. But one of the things I wanted to, from the perspective of my personal research program, one of the reasons collections are really important to me is that they enable me to access and think about some of the big patterns of biological diversity. And one of the things that I work on in my research is why there is so much variation in species diversity around the world. So when you look at the tropics and the temperate zone, for example, why we have these diversity gradients, latitudinal diversity gradients, and so on. So it's just a heat map of global lizard and snake diversity. And I think one of the things you see quickly when you start looking at the data that we have to bear on these kinds of patterns is that this is just the corresponding plot of the data density that we have from GBIF for reptile diversity worldwide. So you can contrast the data density in the center to the species diversity that we have on the left. And the interesting thing that you can see is that the places where most of the species are, like the Amazon, are the places where we have the least amount of data. And so one of the points that I want to make here is that when we think about these big patterns, like why are there so many species in the tropics? If you take any given species of reptile, like this Tenotus skink that I collected in Western Australia a few years ago, Tenotus uber, to answer these questions about why there are so many species in places like Australia or the Amazon, we have to know the answers to basic questions like what habitats do they use? What do they eat? How abundant are they? How do their abundance change through time? How do these species interact with predators and parasites and prey? How is their population structured in space and so on? But I would contend that we lack even the most basic data to solve some of these greatest mysteries of biological diversity. So we don't have the answers to most of these questions for most of the species on Earth. And this is a nice paper from a few years ago that really highlights these kinds of biodiversity knowledge shortfalls that we have, ranging from taxonomic inadequacy to lack of knowledge of species distributions, lack of knowledge of basic ecological traits, lack of knowledge of evolutionary potential, lack of knowledge of ecological interactions, and how profoundly limiting all of these shortfalls are for what we can say about what structures biodiversity in space and time. Now, I want to add one shortfall to this, and it's sort of the shortfall of shortfalls, because my contention is that the data that we need are actually being lost forever, and they're being lost at an unprecedented rate, and that natural history collections are really the key to understanding large-scale patterns and global change. And when we think about places like the Amazon, which are undergoing rapid deforestation, and we think about all the other indicators that we have for change on a global scale, the one thing that really keeps me up at night and disturbs me profoundly is that what we're really looking at is, in light of all of these shortfalls that we already have in addressing these basic problems in the distribution of species diversity in space and time, we're facing an irreversible loss of biodiversity data. And that loss is happening right now at a rate that vastly exceeds the loss of the species themselves. So our ability to answer questions about what shapes species diversity in space and time is, from my perspective, degrading very rapidly. It's degrading much more rapidly than the rate at which species themselves are going extinct. And so when we look at these, trays of museum specimens, these are some specimens from our own fieldwork. We look at what the modern collection process entails, and we generate these high-resolution snapshots of biodiversity, multidimensional biodiversity in space and time. And I think that when we go to the field today, we take genetic resources, we take things that let us track disease dynamics, we take parasite samples, we take microbiome samples, we take skin samples and venom samples and other things that we can use for chemical ecology. We record quantitative habitat activity and behavioral data that can be linked to specimens that we have in our collections. And I think that a lot of folks look at museum collections and ask, what can museum collections do for me? And I guess there's almost this attitude that we can just call up a museum collection and place an order for a set of herpetology specimens or whatever. And I think I just want to call attention to the fact that all of these specimens also come from really hard, boots in the mud field work, and that I think that addressing many of the most pressing challenges that we face requires a renewed commitment to collecting and archiving new biodiversity data. And that's something that I hope does not get lost in this recognition that specimens can provide lots of information for us as we move forward. So there's a huge and acute need for actually undertaking large expeditions today and getting back to these fundamentals of going to the field and collecting fundamental biodiversity data and creating these permanent records of biodiversity in space and time. And just to sort of finish this up, this is a wonderful paper led by Cody Thompson from UMMZ Mammals, just published earlier this year. And the title of this paper is "Preserve a Voucher Specimen, the Critical Need for Integrating Natural History Collections in Infectious Disease Studies." And I just want to add to the title, which I think is almost perfect. And I'd say, preserve a voucher specimen is really key here. And I would say this is critical for the very future of ecology and evolution and global change biology and more. And so with that, I'm gonna just introduce our speaker for today, Professor Guralnick. Sorry, a little delay in unsharing my screen here. So Our speaker for today is Professor Rob Gerolnick, who's a professor and curator at the University of Florida and in the Florida Museum of Natural History. And Rob had his PhD from Berkeley, and he worked on nearshore mollusks with David Lindbergh. And so he has a solid organismal background, and he moved from there to a faculty position at the University of Colorado, where he was also a curator of invertebrates. And since that time, he's moved to his present position at the University of Florida. And it's really hard to imagine, and it's actually very hard to summarize Rob's accomplishments, and especially the breadth of his research in any sort of simple fashion. I mean, he's worked on everything from on the empirical side, from population genomics, remote sensing, actual acquisition of biodiversity data, ancient DNA, niche modeling, conservation biology, life history evolution. It's just an incredible string of like themes that all relate to various facets of biodiversity. But I think the place that Rob has really shown incredible leadership is in the biodiversity informatics front. And he's been a leader in thinking about the very data that we put in biodiversity databases. What are our standards? How do we make the data available? How do we use remote sensing technologies to gain more of that data? So he's a pioneer in thinking about biodiversity databases, automation, tool development, web-based tool development, all these sorts of things. So it's really hard to imagine in some ways where the modern field of biodiversity informatics would be without Rob's leadership and vision in this sense. So I won't continue on here, but I'll turn this over to Rob at this point, who's going to tell us something about new uses for natural history collections. So thanks.
Hey, Dan, thanks a lot. That was a great introduction. I loved hearing about the importance of field collecting. I totally agree with a lot of the great comments you made to start this symposium. I want to say hi to everybody as well. Hopefully you can all hear me and everything is good. It's really wonderful to be here today to talk about some of the work that I do using natural history collections and collections data. I'm also super excited to also catch talks over the next few Fridays from a new generation of scientists, showcasing innovative use of natural history collections to address key scientific and societal issues. So one last thing before I jump in here, I always start my talks with this sort of contemplative statue that captures the idea of balance, because I want to cover topics that balance across data sciences and domain side work, mostly in global change biology and biogeography. This contemplative statue is also a reminder for me to take it easy and slow down. I tend to be a pretty fast talker, so I'll try to keep it slow, and the slide is my guiding slide, and remember to slow it down and keep it slow. Okay, so jumping right in here, what I want to do is take you on a journey that really started when I arrived to the University of Florida 6 years ago. And like so many journeys, it wasn't one I totally expected to take. It hasn't necessarily led me to places I thought I would get to, or has led to those places. And reflecting back on it, it connects a lot of my broader concerns as a scientist. How to thread together data sciences, the sort of concept of the extended specimen, which I'll cover later in this talk, and especially the importance of traits and how they are measured on specimens and how they're used to answer questions. Okay, so I think the thing that really catalyzed this journey in a lot of respects started with a quote from a colleague now at UF about trait databases. And I'll just summarize the key points here, since you can read the quote yourself, but I want to drive home the key message here. And that's really about the fact that most trait databases are summarizing data at the species level, species level mean values. And that's a good starting place, but I think the important thing here is to really understand how traits vary across environments. We really need to think about measuring traits at the individual level, not at the species level. And so I started to wonder how we could assemble our knowledge of traits at the individual level. And I was especially interested in integrator traits like body size and body mass, because body mass in particular, well, body size in general, plays a really key role in evolutionary and ecological studies. So for many years, I've been really fortunate to be involved with a project called VertNet that many of you probably know. And I noticed that trait data sometimes is included in published records on VertNet. Usually the measurements are in weird notations or have challenging abbreviations, but they often are really rich. So here is a VertNet specimen record of a wood rat. And here you can see on the label, right, some information about commonly taken external measurements that were probably, in the shorthand, format that was probably from like the big dawn of mammalogy. The Seahart Merriam or some mammologist said, report the traits like this on labels. So we have like a total length, and we have some external measurements like hind foot and tail length. And we have some measurements of the total mass in grams. But those measurements are really, really important and valuable. But they're often really hidden. So let's take a look at that in a little more detail here. So if we think about a record from this red squirrel that is available in Arctos, we can see, well, wow, we can see that this specimen was collected with a firearm. Okay, yeah. But in this odd field, when we represent these data in Darwin core format, which is this really commonly used standard, if we look deeper in this record, we can see that there's actually some really important information about traits in here. So we can see that this specimen had a hind foot with its claw on that had a length of 49 millimeters, and we can see these total length and mass measurements. So while this isn't in a standardized format, it's in Darwin core, there is no Darwin core field that says hind foot with claw, right? This data is hidden in the record and impossible to really recover easily from across the whole corpus of mammal records in VertNet, right? So the idea would be like, could we standardize the data on body mass and total body length in a way that it could be represented as harmonized, so it was discoverable across this whole data set? And so that is what we've decided to take on. And I don't want to go into the gory details of this, but I thought that maybe we could do something here. Maybe we could harmonize all this trait data. So when I got to UF, the first person who I brought on board was a software developer who had a passion for natural language processing and text mining. And I think in the case of these records, it may have been more like unnatural language processing. But anyway, we worked together with colleagues, John Wieczorek and Paula Zermaglio, Paula shown here on the upper right of the slide, to work out a little recipe, right? It was a recipe for getting these data out. So what we did was we took 20 million digitized vertebrate specimen records. We decomposed those records to look for these hidden traits and measurement information. We then extracted records that matched an exhaustive set of synonymies that we developed. And then we harmonized as best we could to standardize units. And so the synonymies were basically those abbreviations, those sort of weird formats and notations that people use for these trait data to get those together. And we did a lot of curation to validate our automated extractions, like a lot. So we wanted to really be sure that we could reduce our false positive and false negative rates so that they were super low. We didn't want to have traits that we missed, and we didn't want to have false positives, cases where we extracted something that wasn't actually a trait. And so after we went through this whole process of refining this and tweaking this tool set to basically deliver the best possible trait data out, which my developer Rafe referred to as whacking moles, to sort of you solve one problem, another one pops up. But after we finally got that to the point where we were satisfied with it, we were able to kind of serve it back out and make it this delicious meal that we could serve back to our community. And we also wanted to assure anyone else could also use that toolkit to reproduce the work or to use it for new work. And so we developed a whole package called Trader that allows people to kind of use these trait extraction toolkits themselves. And once we were done, we kind of wanted to know, well, what does it really tell us about how much trait data is available as it stands now? And here we were explicitly after kind of two key measurements, total length and body mass, no other trait measurements. And I think the answer here is both wonderful and terrible. It's wonderful because we got a lot of trait data extracted. Let's just take a look on for aves, for birds and for mammals. And no, Dan, no disrespect at all to the herp and Ix people. It's just that down the road in the talk, I'll be using the bird and mammal data for a couple of example studies. What we can see here is we have a lot of species where there are at least 100 length or mass measurements. Like for mammals, and in terms of length, we have like 417 species where there are at least 100 length measurements for those species, which is really great. It's also a little bit terrible Because this is still just a fraction of the data that we know is still out there on labels or even digitized and just not yet available on our specimen data sharing networks, right? So this is like the tip of the iceberg on what we can do in terms of the trait data. So we also did another thing that I think was really important, and I'm going to come back to it at the end of this talk. We also assembled at least some information on the traits we didn't extract. So this is like a bubble plot that's showing you the overall numbers of these trait types. We kind of typify the traits. We want to know the general number of beak traits, head traits, appendage traits, trunk traits. And yeah, so you can see there's a lot of gonad traits and a lot of beak measurements. But those are really awesome measurements to have. I mean, so much life history revolves around reproduction. And so like knowing that we have all these other traits that we could go after later is super cool. This is like a universe of traits that we can go and work on downstream. Okay, cool. So then we thought to ourselves, we should really just get these data back out to people. That came from community effort. We wanted to push it back out to the community as fast as we possibly could. So what we did was we basically indexed these data and made these data immediately available back in the VertNet portal. So we did this well before we started publishing any work on body size, because we felt the faster the better for sharing, for increasing creativity. There's lots of people who might want to use these data in ways that we haven't even thought of. Get it out there, let them search for it. So then, VertNet portal now has a way you can, filter for records that have a length or a mass, and you can query these, in all kinds of neat ways, right? Which is really nice. And we wanted to tell people what we did. We wanted to let them know and be able to find the data and see it. And we wanted to share the knowledge and get the word out there. And that's exactly what we did. And it kind of ends the first step in this journey. So that ends step one. But I realize this next slide sounds a little like a greeting card. And so apologies, but actually it was serendipity that was the next step in this journey. So I had long been thinking about how to use these trait data at scale. And I ran in randomly to a colleague and a graduate student who were interested in data-intensive questions about body size. And basically the idea was to use data from these, hundreds or maybe even thousands of species to examine this very old and still contentious idea called Bourbon's rule. And so Bourbon's rule is a rule that was proposed long ago, back in 1847, and is really thought to apply both at intra and interspecific levels. And when Bourbon proposed this rule, he didn't have an explanation at heart behind how it worked, which is related to heat loss, and how this relates to body size. So, I mean, you can think about this purely and simply from maybe simple examples of cube animals and their surface to volume ratios of these larger and smaller cube animals shown on the bottom right and the top left. And so basically, the smaller cube animal has a much higher surface to volume ratio than the larger one. And because of heat loss around the surfaces, the idea was basically that larger animals are going to have reduced heat loss, and so they're going to be able to be found in maybe these colder, higher latitude areas. And so if you look at this sort of classic relationship for Bourbon's rule, what you can see here is basically that larger animals within a species might be found in these colder environments, and smaller animals are found in these warmer environments, right? Around this idea about heat loss. But it turns out that Bourbon's rule is really contentious. There's lots of conceptual concerns about Bourbon's. It isn't clear if the rule is a pattern, if it has a fully articulated mechanism, whether those mechanisms are the same at the intra and interspecific level, and whether it's really broadly applicable across animals or more narrowly applicable only in homeotherms. I can go on about these different arguments, but it's also one that I think has some concerns conceptually. And so conceptually, I should say empirically, there are some really important questions about whether this is due to publication bias. So we might think about the fact that if people aren't publishing results that don't support Bourbon's or that are non-significant. And there's been really, really different ways that people have done studies about these relationships between climate and body size. So people did measure different things. They use different subpopulations or populations across different scales. And so really understanding how to put this together and look at this beyond a meta analytic approach, it seemingly would be a really, really important idea. So I think there's a real chance to basically use our trait data to take a data intensive look at Bourbon's, right? And that's what we did. So in collaboration with Christina Reamer here, shown on the right, and Ethan White, with Christina leading this work, the game plan was to basically associate body mass values with temperature at the location and year of collection of those records, as well as like actually kind of fitting these potential lags so that the previous year or previous 5 years could be used instead of the year of collection. Then to carefully filter for outliers and remove juveniles, and then basically simply fit these mass temperature relationships for as many species as possible where the data were available. We also wanted to look at some questions long asked in the literature about whether intra-specific Bourbons is stronger for larger or smaller bodied animals, whether it's stronger at higher latitudes or varies by class. And I'm not going to actually cover those, but I'm going to just mention that it turned out that we really didn't find much pattern here in terms of these kind of proposed hypotheses. Nothing popped out as being strongly supportive that Bourbons was differential across these different kinds of parameter spaces. And let's take a look at how the data sort of shape up at the global scale. So each one of these black dots on a map is actually a report of a body mass that we derive from this VertNet data that we were talking about earlier. And I pulled forward here 3 species: Martis pinati, the fisher, a chipmunk, Timius quadrivittatus, Synaptimus cooperi, the southern bog lemming. And I wanted to just point out that those 3 species are interesting because they kind of reflect different possibilities, right? We see that classical Bourbon shape, that relationship for the fisher. For our chipmunk, you can see there's really no relationship between mean annual temperature and mass. And then for our southern bog lemming, we see a converse Bourbon's relationship, right? So when we look at this over all the species, we had data to fit models for, which is about 960. You can see that while there might be a slight tendency for more negative relationships than positive ones, the vast majority of species don't have a statistically significant relationship between body mass and temperature. And it doesn't really vary much by clade. So like looking at birds versus mammals. And we've actually followed up on this work quite a bit. I'm not going to present it in detail here, but we fit many more climatic variables. We've incorporated more covariates beyond those climatic variables, like season collected and sex. We've accounted for phylogeny in these analyses using phylogenetically generalized least squares. And we don't see really that much improvement in these models. There are still relatively weak effects. So again, looking at these relationships, we sort of have a now data intensive ability to look at this in a single modeling framework, right? One modeling framework, we're going to ask these kinds of questions and understand more about body mass relationships with climate. Okay, so this really is a bookend for a next step in the journey. And what I thought might be particularly interesting when I started thinking about these climate body size relationships was if we could look at body size change over recent time. So in fact, a lot of the reemergence of Bourbon's rule was in the context of this idea that phenotypic change and body mass shifts might be a third universal response to climate change. I had also wondered if maybe we could look at disturbance, in particular looking at human disturbance and how that might impact body size. But it turns out this is really hard to do. I mean, it shouldn't be necessarily, right? When we have a specimen, it has a date collected, on that specimen. We know when that specimen was found and we have a location. It shouldn't be super hard to look at this in a temporal context, right? But actually, it's really hard to do this because of spatial and temporal biases in these data. They could potentially really limit what we can do here. And so that's a really important thing to think about in terms of understanding change over time. But I think there is basically a databasis for asking some of these questions. Before I jump in here and do this, what I want to do is basically introduce this amazing star of the next half of my talk, which is Perimiscus Mediculitas, which is probably the most common and most commonly trapped mammal in North America. I'm going to refer to the species as Pima. So Pima means Perimiscus Mediculitas, it's just an abbreviation. And this redonkulous animal is really honestly something of a pest in suburban areas and a carrier of some nasty things like hantavirus. But by far and away, the richest numbers of body mass data and link data that we have available are for this species. And this provides us a chance to ask some interesting questions. The rich data for PIMA give us a window into asking some questions. In particular, does human disturbance, and in this case, we're going to think about human disturbance as related to urbanization and human population density, impact mammal body size? And if so, is this a result of basically just simply an urban heat island where urban areas are warmer? Or is there alteration of habitat and food resources that might drive body size change? Or maybe it's actually potentially fragmentation. Fragmented urban areas might actually create islands where body size will respond according to island rules. So those are all really interesting questions that we can begin to ask if we knew how to look at the temporality of the data and think about how that might work. So what we did was we assembled a relatively unique set of data for this. And so before I jump into telling you more about models and some of the results we found, I want to tell you a little bit about the data resources that we used for this study. So we didn't only use natural history collections data, we also pulled two different data resources. One was the North American Census of Small Mammals. This was a census that was done in the 40s and 50s. It generated a ton of data at a continental scale using a consistent survey method. And it's really awesome to go back and recognize just how much people were thinking about surveying broadly, going out and getting those data, collecting those voucher specimens back in the 40s and 50s. And of course, NEON's doing some of the same kinds of data collecting now. NEON has a small mammal trapping data project that they do across their sites. And actually, if you put these data all together, you can see that this is relatively amazing in terms of the data density. It's not like there isn't some increase in data density over time. So there is an increasing amount of data towards the present, but it's not terribly biased. And so you can think about, say, 1950 and how many records were available then versus, say, 1995 or 2015. And it's not too far off the money to think about looking at these data in one go, to think about trends in body size over time. So that's exactly what we did. But to get there, we had to think really, really, really hard about some of the problems with how to localize data across space. I also want to mention before I move on that one of the things that we were surprised to find is that body mass and head body length, which is basically just the length of PIMA from the head to the beginning of the tail. So we can take like total length of the specimen minus the tail length to get this head body length. These weren't highly correlated with each other. The correlations were about 0.3 or 0.4. So we want to think about using both of these measures, body mass and head body length, separately when we think about how to model changes in body size. Okay, so basically to look at this question about body size change, and to examine these trends, we wanted to localize the regions in order to basically have replicated regions where we can examine body size trends across space. And so what we did was we basically built these evenly sized grid cells as spatial replicates. So again, we could localize the effects and use these spatial replicates as random effects in linear mixed models. So to do that, we had to basically think through a couple of optimization challenges. How to optimize the most number of records into non-overlapping grid cells. In this case, we set a rule where we required there to be at least 4 decades where there were at least 10 records of mass or head body length measurements per those 4 decades. And then we also gathered climatic variables and human population density data for each record at the location it was collected. And we were really fortunate that there were recently published decadal estimates of human population density available at relatively fine resolution. So then what we did was we basically fit 2 classes of models. The first one I'm going to cover here is explicitly spatial, but we also fit a temporal model. The spatial model used climate and urbanization as fixed effects, along with season collected and sex. And then we used grid cell and the source of our data, whether it was NACSM or NEON or natural history collections data, as a random effect. And then what we can do is we can basically start looking at some of these patterns. And so for the spatial models, we're going to focus initially, we can look at the trend in terms of body mass in relation to the climate gradient, looking at mean annual temperature or mean annual precipitation. And we can see for both body mass and head body length for mean annual temperature, we see relationships that look like Bourbon's, where we have the lower body masses and warmer conditions. And then for precipitation, we can see, sort of surprising to me, which is not the usual way that this relationship works. Actually, the smaller Pima in more wet areas, right? So we also wanted to look at the relationships for body size in relationship to human population density. And here we can see just for head body length, not for body mass, the head body length decreases with human population density increases. And we also, just to kind of show you some of the covariates that we fit as sort of to control for these effects, we also can see here some of these relationships between sex and season. Okay, so finally, we wanted to fit these temporal model. Sorry. Let me go back a step. So we did one other thing. And this is really important. I'm glad I kind of pushed this in here. We fit source as a random effect in our models, and we're really glad we did this. And here's why. We don't see a strong effect of source for body mass. Those are pretty much the same body masses across NEON data, ECSS data, and natural history collections data. But we found a really different pattern for head body length. And here, NEON data were always significantly smaller than the data from NCSM and from the museums. And we thought at first this might be a real effect related to time. We went over and talked to our friends in NEON, and what we learned was that since these measurements were always taken on live animals rather than dead ones, and it turns out NCSM didn't live trap. It's very difficult to actually measure the length of a live mammal. So while mass is comparable, you can put the mammal in a bag and measure it. Length is really hard to capture, and it's always underestimated because of the way it's measured. So it's not that there's an actual trend here in terms of NEON data, which are more recently collected. It's really a bias in these data that we didn't know about. And so if we didn't carefully think about that sort of bias, it would have really impacted our model results. So clearly we did not include NEON data in our final head body based models. So let me just show you the models here that cover the temporal trends. So this is really super interesting. I want to draw your eye towards the results for body mass, the body mass temporal model. And what we see here is that basically this model shows us that body mass is declining, is decreasing as we go forward in time, as we use decade as a covariate, right? I'm over here in this decade minus. That's just showing us that basically there's a negative relationship between decade and body mass. However, we tested this really carefully by looking at the delta change in temperature, urbanization, and size within each of our grid cells, using those decade-based slopes that we were able to generate from our random effects. And we really didn't see any indication that increasing urbanization or climate was strongly explanatory of body size change. And so that's really super interesting. It's sort of a puzzle. We have basically this indication that Pima are getting smaller in terms of their mass over time, not their length, just their mass. But we're not quite sure what the driver is on that. So I think the take homes on this really are that, different size proxies might show different patterns of response spatially and temporally. Deer mice are basically shorter but not slimmer in urbanized areas or human population dense areas. This might reflect the heat island, but it might potentially also be related to crypsis. And so it's possible that being small avoids detection by humans, and this could be an advantage. And we do find that Pima mass but not length is decreasing over time. This isn't in relation to urbanization and climate. But okay, so one species, just this one very well sampled species. A bigger question is how response varies across species with different life histories. So we started to think about this again, and we thought maybe we can go bigger and really look at this over a larger set of species and put it all into one modeling framework. So I don't want to spend too much time on this because I want to move on to a couple other things before I run out of time here. But I want to call out Maggie Hancock, a PRFB postdoc in my lab who predominantly works on herps, but has deigned to work on a couple mammal projects. And Maggie has been working on looking at the same general question we looked at in Pima, but using a multi-species approach where we can collate traits such as average body size or thermal buffering capacity, diurnality, to see if species with different traits are showing different responses to climate and urbanization. So what we did was we brought together over 140,000 body size measurements over hundreds of species and greater than an 80-year time span to look at these questions. And what I don't want to do is I don't want to take too much time here to go over this in great detail, but I do want to just cover a couple of the things here that are of interest. So this is more like a taster, but we were basically able to understand more from this sort of look across species and across space and time at drivers. And in particular, we find that urbanization conditions this temperature body size response with much stronger responses, stronger slopes in areas with with high human population density. So we see a stronger relationship between body mass and temperature in these human dense areas. And the slopes are much weaker and shallower for areas that are more in wildland areas where humans aren't, which is super fascinating. And it's kind of hard to see in these lower panels, but if we split mammals into groupings of large and small body-sized, average body-sized mammals, large mammals are actually increasing in length across the gradient in terms of human population density, while small mammals show a very, very weak decrease. So interestingly, this doesn't really align very well with either an island rural effect of urbanization, nor does it really support the idea that urban heat islands are the core drivers of these responses. Instead, we do think this might provide some support, and I can explain a little more why if you're interested in the panel discussions or afterwards, for the idea that maybe resource allocations are driving some of these questions about body size relationships across the gradient of where humans are found. We can also see that there is a sort of interestingly different kind of pattern for mean annual temperature in relationship to temperature and body mass along the gradient for larger and smaller mammals. This gives us a lot of power to look at these kinds of questions in a joint modeling framework, in a single modeling framework. And that kind of leads me to my main point here, which is that rather than like kind of boring with more charts and graphs, I guess the take home is that we can start building broader frameworks to understand phenotypic response using natural history collections data. And to me, the biggest surprise of all of this work is that urbanization effects for body size appear to be really super strong, perhaps rivaling climate factors long thought to be really critically important for conditioning body size of mammals. So this is like super fascinating and an area we'll be continuing to explore in some detail in the future. Because really these are very broad patterns and we want to kind of get to the more narrow drivers and really understand the mechanisms behind some of these results. Okay, I have one more data vignette to show you. And I'm going to close by bringing it back to data sciences. And so we walked along this path. We walked down the road of looking at these body size relationships. But I wanted to take one more sort of sidestep towards another area of interest of mine that has grown over the years in terms of phenology. And this is going to be more broad brush than it deserves, but I think phenology is really important and really critical because when we think about those universal responses to climate change or global change, we're thinking about species distributions, we're thinking about phenology, and we're thinking about phenotype changes, right? So we're going to cover phenology really quickly here. Okay. So what I want to really do here is just basically show you that we can use natural history collections data to look at phenology climate relationships at much broader scales than we've ever been able to see before. And again, I'm going to use PIMA as a test case. And I'm also want to point out that this is really fun collaborative work with Brian McLean, a mammologist who was a PRFB, NSF PRFB fellow at Florida with me, and now is a faculty member at UNC. So how do we collect data for this study? Well, actually, in this case, we were able to use a tremendous amount of information from specimen records reporting reproductive condition. We also gathered reproductive state information from museum specimen skin tags, along with direct examination of specimens for secondary characters that are indicative of breeding. So on your labels, you'll often see reports of that a female is reproductive, it had embryos, and it was lactating, it was pregnant. And you can also use these sort of secondary breeding characteristics to basically detect on the specimen itself whether the female was potentially breeding. Finally, we also pulled published reproductive state observations contained again in the NACSM, in that census of small mammals, and also in the NEON data that captures reproductive condition as well. What we did was we used those presences and absences of reproduction in basically a hierarchical general additive modeling framework that was constrained to be cyclical to simply determine basically the shape of phenologies over the year. So you can see day of year on the bottom part of these plots on the right, and on the y-axis you see the probability of breeding. And it's really gratifying because what these show are things that are really intuitive to people, and you kind of go, oh, I got that. Like northern forests up in the north, you see these shorter phenology periods that are peaked pretty strongly, where you have high probabilities of breeding in late spring. You have really low probabilities of breeding during the winter period. But like in the Mediterranean, California ecoregion, where we kind of fit these models across these ecoregions using these hierarchical approaches, you can see like in California, it's like a really temperate, mild climate. Pima are breeding year-round effectively at lower probabilities, right? Which is really cool. And we can also see some things we didn't expect. We can see some humped, bimodal distributions where we see two peaks be reflecting low probability of breeding, especially in summer months where it might be too warm. So in general, what's really cool is we can kind of see things that make sense to us. We also did the same kind of analysis thinking about not ecoregions, but basically spatially separatable climate clusters, areas that are climatically different that are spatially arranged across the continent. And we basically used a spatial clustering, accommodative spatial clustering mechanisms with climate data to kind of pull out those clusters and look at phenology across those climate clusters, right? And then what we could do is we could basically use linear mixed models using temperature and precipitation of the prior months from the collection date of the sample, along with photo period as variables, as covariates, to basically understand what climate predictors are important for driving phenology. And I'm not going to go into excruciating detail here, because you can read about this work in this month's issue of Ecology, where the paper is now out. But what we also did was we basically extracted these slope estimates for the random effects across these climate clusters to see if there were stronger or weaker relationships in different climate regions in terms of the relationship between breeding and, say, temperature or precipitation. And what this plot on the right shows is that both temperature, seasonality, and precipitation both seem to strongly condition the strength of Pima phenological responses to either precipitation or temperature as sort of a short-term resource availability axis. And though I might be a little bit skeptical about how we can do space for time substitutions in relationship to understanding how this might relate to breeding responses over time, we're super interested to go back and see if we can pick up, basically a phenological signal over time in the data we have, to understand if there is a way that we can use these spatial responses and maybe even the temporal responses to predict response of reproductive phenologies in the future, right? Because so we can do that, we're basically able to get ahead of understanding how phenology might change in response to changing global conditions. Okay, so phew, we made it to near the end of this talk. So what I want to do in the very last part of this talk, and this is last but not least, and pretty quickly, is coming back to where we started about data sciences. And so this is my way of looping the talk, and so the footprints of my journey come full circle, right? So I mentioned we pulled total length and body mass as two key traits, but I actually used in a couple of the studies I presented head body length. And you might be wondering to yourself, well, how did you get that measurement? Like, you had the total lengths, but what about the tails? How did you get that just the head body size measurements? So to a larger point, there are huge numbers of external measurements people take on specimens. And there are even more numbers of anatomical measurements that are taken by people, especially given all the new tools we have to measure phenotypes. And Dan mentioned CT scanning. It's amazing. I'm really impressed by the work we're doing at the Florida Museum and broadly on that topic as part of OVERT. So there's all this new measurement data that we're going to be able to generate on specimens. And we also have a ton of different resources. I mentioned VertNet, and we pull data out of VertNet, but there's lots of different resources out there. It's not just VertNet. There's also, basically all these other resources that span from archaeology to paleontology, where measurement sits critically in much of what paleontologists think about and do, all the way through to resources that may have trait data available that just don't even know it yet. So there's all these other resources, intentionally or not, that cross multiple disciplines that sort trait measurements. So the question I had is, how are we going to build a better mousetrap for making trait data available, right? And so that is a question that I was really fascinated by. And we were lucky enough to receive some funding from NSF to try to build a new type of trait data store explicitly focused on measurements taken from individuals and spanning across neontology, archaeology, and paleontology. We call this project Futurus, the functional trait resource for environmental studies. And it's really a project that at heart thinks about how we develop communities and best practices around storing, aggregating, assembling, using measurement data from specimens. So providers can use a set of tools to mobilize their trait data into Futurus, where we can help to reconcile how people measure traits and standardize that reporting, clean the data, and actually use machine reasoning to make inferences over that data to help with discoverability. And then we provide ways for users to access all that content. I'm not going to go over all the details. I want to be cognizant of my time, but Futurus can help assure data reuse. And I think most excitingly, it can be used for things like allometry studies, where if we have a lot of mass estimates taken from modern animals, and we have a lot of anatomical measurements taken across archaeo and paleo, we can begin to improve our allometric equations and understand how to use them more effectively in the context of these broadly and simple kind of data sets. So behind Futurus is a trait ontology. I'm not going to bore you with the details, but the key thing is that backing Futurus is basically the definitions of traits and their measurements. And that's done as a community process where people have traits that they want to bring to Futurus. We go, okay, yeah, we don't have that trait in our trait ontology yet, but we're going to define it. We're going to define it in the way that the community can access those trait definitions, via the interwebs using URIs. And they're going to be tied to other existing ontologies to describe anatomy, like Uberon and Pato for traits. So that's exciting because it means that we basically have this sort of ontological and semantic framework and linked open data framework that sits at the heart of what Futurus is all about. And so just this last month, we were able to release our beta portal, where you can actually go and find data about body mass and body lengths of all sorts. You can see that the traits that are available are much more than just length or mass, or at least body length and mass. So there's all kinds of data available here, and you can filter this really easily by life stage and by trait, by taxon, or by project, and get a really awesome set of data that you can use for your own studies. So check it out. Okay, so I've come to the end of my journey. Actually, I have one more thing to talk about very quickly at the end here. And I just want to say that one of the most gratifying things about the work shown here is that, besides following my feet and oftentimes following with my friends and colleagues and making that trail, many others have seen the power of what we now call the extended specimen to fuel new research. And it's really an exciting time to be working on natural history and with natural history collections data. And whether it's genomics or phenomics, or whether we're talking about species interactions, or working with the specimens and that relate back to the environment, our ability to take those, bring them out and work with them, and then look at how that relates to broad scale questions that Dan brilliantly talked about at the beginning of his introduction, is really, really powerful and really, really exciting and gratifying. Funnily enough, this slide is actually from a 2014 talk I gave. It was the last slide of a presentation, kind of conceptualizing my view of what the extended specimen network may look like over time. And it's fun to put it into a talk, 7 years later, and realize it's just as relevant, just as interesting, and even more exciting now than it was when I first was thinking about this back in 2014. And with that, I have a huge host of acknowledgements to make really quickly here. This journey was one that was taken with lots of people: Paulo Zermaglio, Narani Barvi, Brian McLean, John Machorik, Daejeon Lee, who is an assistant professor of quantitative ecology now at LSU, Maggie Hancock, with whose main interests are in polymorphisms and trait evolution, and our great futures postdoc Megan Balk, who's really interested in body size eco-evo questions. And finally, Marie-Fleur France, who is amazingly hard to photograph, who's this incredible informatics developer, has been key for a lot of this work. And with that, I just want to say thank you guys so much for listening. I hope I managed to make it through the end without losing my voice or having the microphone give out, but I'm really excited to be taking part in the panel coming up here. Thank you very much.
Well, thank you, Rob. I guess we'll give you the virtual applause. So thanks for the great talk. It was fantastic. Just a reminder to everybody, if you have questions for Rob or Dan as we kind of work into the panel discussion, please use the Q&A box and we'll work through those questions as they come up. And and if you would, please address them if you would like them to go to Rob or Dan specifically, and we'll address them that way. Rob, there's a couple questions already in the Q&A that I'll start with that are, one is definitely tied to some of your research. And it's from Holly Shear. How and why do you filter out large animals from your body size study? What is the significance of outliers? So it's a two-part question.
So we've been really concerned about a problem with some of the data that we have from VertNet. And so in the vast majority of cases, we don't have a life history stage reported for the records. So we don't know if they're adults or juveniles. And so because of that problem, it's really difficult to sort of not befitting kind of ontogenetic change of body size. If it turns out that there are biases in juveniles and adults across space or time, we wanted to kind of try to eliminate that problem. And one of the ways we thought about doing this is, we do have some reports of juvenile versus adults. So we can kind of create distributions of the shapes of the juveniles versus the adults and use those distributions to help us make predictions about how to filter those records that don't have those reports of juveniles and adults on them. And so there are a bunch of different methods that we've used to sort of try to filter records to be usable. But one of the really interesting things about that is, the first thing we thought of, well, we're just going to use, basically the great work that's happened mostly in Michigan, I think, on ADW to kind of get the adult body size range. But when we just purely kind of cropped the body mass distributions to the range that are reported in the literature, it does a couple things. First of all, it assumes the literature is right, which it probably isn't. And second of all, it tends to skew your distributions to look funny when you think about them in a statistical framework. And so we've been thinking about this one forever. And what I like to really encourage is, as we get better and better at publishing our records about the specimens we have, incorporating like life stage and life history information with those records is super valuable for their best use downstream for global change studies. I don't know, did that actually answer your question in the right way?
Let's see if Polly will add a comment here, but I think that sounds great, Rob. Kind of to build off of that, one of the questions from the panelists or the ECSS committee that I think relates to that question is that taxonomy on these obviously is an important underpinning of your work, and making sure that the labels on the tags are what they are, right? As a mammologist myself, your work with Perimiscus Mediculitas, probably one of the most misidentified animals in North America, despite the fact that it's probably one of the most abundant mammals in North America. And there's other things. There's likely that there's more than one species there. How do you guys account for that in your trait work when you're really kind of getting down to the nitty gritty of very subtle differences in mass and measurement data that's taken across a broad range of this particular species?
Yeah, great question, Cody. So I think like for a long time, in my view, taxonomy was like the third rail of biodiversity informatics. It's so difficult, right? And at the heart of the taxonomy problem is two different two different issues, three different issues. Some a little more trivial, some really, really complicated. The first issue that I think is less complicated is basically linking up names around synonymy, currently valid names versus names that are not currently valid, that are out of date. And that problem is pretty easily solvable. The second really much less soluble problem is taxon concepts. Do we have the delimitations right? And as new information is generated that says, this is actually a split, or we're recombining, basically two name units into one via lumping, how can we, basically get that information out fast enough? And so like for herps, like the split rates are ridiculous. Like I think if you look at the flux of concepts in like in neurons, it's incredible, right? In like 20 years, I think like 15 to 20% of the names have changed, which is a really huge amount of change that needs to be linked back to the specimens, right? Because the specimens bear those names, and and those labels need to be updated to reflect the current knowledge. The least soluble problem from my perspective is the hardest one of all, and it's one you've mentioned, Cody, and it keeps me up at night. Dan gets kept up at night about stuff. I have the same things that keep me up at night as Dan does, but I also get kept up at night by misidentifications. And really we're just gonna have to rely on the fact that as more of these data get used, we have to go back to the collections. For the phenology study, Brian McLean, I hope he's actually here on this call, this symposium, and I were at Smithsonian looking at these specimens and recognizing that some of the work just has to be back in the museums. Brian says here, awesome. Brian, if you want to jump in on the PIMA misidentification rate, you're welcome to in chat. But I think that this is a larger problem, and I think if you talk to anyone across any collection in the museums, and Brad Ruffel, I'm thinking about at the Florida Museum, Walt's constantly talking about the fact that 30 to 40% of plant specimens might be misidentified. That's a huge problem, and it isn't solvable by doing data sciences. It's solvable by having experts working in taxonomic disciplines. I wanted to mention one really important thing about that. While I will talk about the importance of biodiversity informatics has no value unless it's tied and coupled deeply and strongly to taxon expertise and field-based biology and sort of people working who know their stuff about the specimens that they're working on and the taxa they're working on. So, we need to have a healthy ecosystem, and it's not just that they're going to be data users and people who just blithely use these data. We need to basically remember that you have to have expertise and knowledge and value that in this ecosystem we're going to build that will allow people to do more work with these natural history collections data and innovate with them.
Looks like Dan has a comment there. Go ahead, Dan.
Well, I just want to build on one of Rob's points here, because this just sort of puts me on one of my many soapboxes about various things. But this particular issue about pointing out this when you mentioned frog taxonomy, for example, and this massive change in taxon names, I would just add to that this problem is exponentially worse for any type of biodiversity data that does not involve vouchered specimens. So I try to look at these data sets that people have collected and invested in terms of long-term biodiversity monitoring over 50 years in places like Australia, where I've done a fair amount of work and looked into some of their long-term data sets. And you could not go back through and figure out what a lot of these things are. And it's not just a matter of like, oh, we know that the taxonomy changed at this point in time, so we can flip in a new name. There are fundamental issues, and you're going to say, well, species distributions have changed. In some cases, there are actually legitimate cryptic taxa that are there. I mean, they may not be truly cryptic, but from the perspective of the knowledge that was there 20 years ago, they truly are. They are cryptic. And so you could say, what are we looking at? Like, some taxa are totally unambiguous. There is no question that 50% or more of the species that are named like that are going to be in those types of databases. We could figure out what they are, but when you're not vouchering, you are still looking at like what 15%, 20%, 30%. In some cases, for some taxa, even some vertebrates, at least reptiles and amphibians, would be junk data in some of these databases without voucher specimens. So I think it's a serious issue, and you always have the opportunity, at least with the specimens in this sense, to even with these taxonomic uncertainties, you can go back to things and potentially address some of these, at least with an investment in resources and allocating resources to the specimens you already have.
Hey Dan, I'm gonna double down on that a little more. I feel like we're gonna like continue to like amplify this message a little bit. But one of the reasons why we were so psyched about Futurists is that, you know, the species level trait databases that have like averages, what happens when names change? I mean, what do you do with the trait data? You can't, I mean, do you split it up in half? Do you like, you know, take that mass range that was between 100 and 150 grams and like, you know, put 100 to 125 in one part of the split? And so how do you even do that? If you have the individual measurements, you never have, you know, as long as the identifications are right, which is a huge issue, you can basically parcel out the reporting in the right way from the individuals up. And that's one reason why I think working from specimens and from vouchered specimens is always the core of any kind of biology. So, I mean, not every kind, I mean, yeah, you can remote sense things, but like vast majorities of our knowledge about biodiversity and organismal biodiversity comes from vouchers at heart.
Great. I think moving kind of a related sort of topic, and well, I suspect we'll probably come back to this again. Is, you know, obviously this amount of curation, whether it's curating the labels to current taxonomy, or you know, making sure the data is as high quality as it can be when it goes into a data aggregator and it gets harvested by users. You know, there's an incredible amount of work that happens at the curatorial level, at the collection manager level, that is definitely academic in nature and providing some research perspective to those specimens that are being utilized downstream by other users. How do we, you know, how do we get better about recognizing that work and you know, recognizing the curatorial aspect that goes into managing large collections? I mean, not just large, any collection of any size that are being utilized by the research community.
Cody, that's a great question. And the short answer is that credit models in academia are one of the hardest nuts to crack in all of our problem spaces. I mean, I honestly think that, you know, you know, back 75 years ago, somebody decided, you know, we're going to start counting publications, and that's our unit of currency for what we think about when we talk about, you know, academic value. And I think in 75 years, the world has radically changed. You know, we don't just have to count, you know, how many times a reference in a paper links back to, you know, the document. And I'd say, like, you know, what we need to build is a better way to track what we do professionally that's broader and more interesting than what we have right now. And so I think, you know, there are a couple of you know, sort of promising signs. I know Dave Shorthouse has developed a way to kind of track some of these via a tool called Bloodhound, to track some of these ways that people, you know, make corrections, and then those annotations become publications, or the little micro publications, right? And that can be counted. And it's funny because, you know, we wrote a couple papers on this topic many years ago about kind of micro publications and some of the ways that we could be more flexible about sort of trying to be egalitarian and more openly thinking about capturing more than just your some worth as a person or a researcher via publication trails. And you know, I think that maybe as academic units reevaluate what they really care about and want to find people who are basically committed to broader visions than the next publication, we're going to get there. So like at the Florida Museum, we're actively asking ourselves, is that the only criterion? What about building a better community, building a more diverse and inclusive community? How can we do that and make sure that that becomes part of what we credit in terms of our professional success? And I think we just need to have those conversations and recognize how we do that. I also would say that museums in particular is a little less true of departments. I think intimately, I mean, my museum, I think intimately understands that there are different pieces of the puzzle we need to solve. And I'm not sure it's true to every museum, but I think if museums make the case that they ultimately are one of the most important resources for knowledge of the natural world, and really are firmly taking their place at the table and say, we really have something that is absolutely invaluable. I think we need to see the message louder and then not be the tail wagging the dog on credit models. I think about this a lot because Dan mentioned something really important in his opening presentation, and I think I want to mention one thing about that. And the world has effectively entered a period where it's not only where we're losing the biodiversity, but disequilibrium dynamics are that is a present and future. It's a disequilibrium future. What that means is when we think about climate distribution, species abundance distribution relationships, the data collected today is actually going to be less valuable than the data collected 100 years ago, because we're collecting data about disequilibrium rather than equilibrium that is necessary to understand those relationships. And so our past data will become vastly more important as our future data becomes vastly less about the sort of natural systems components and more about the Anthropocene. And so just a way of saying, a long story short, better models, better social practice. We got to just change our social practices today, not tomorrow, not in 5 years.
Dan?
So I have a follow-up to that, which is, this idea, the point kind of originated with this idea of, in some ways, a credit with respect to curatorial sorts of things and what happens at this level. But there's this flip side of this, which involves what I perceive as a deep pathology of the way science is practiced in some ways, which is that this is not necessarily a fault of the field, but we are a field of biodiversity consumers, and that we consume biodiversity data. And in general, when you play in the sort of intellectual sphere of doing the big questions, we're going to do the big thing in species diversity. We're going to look at speciation rates in a global sense. We're going to do these big macroevolutionary trait things. We're going to map out species and do conservation priorities things and get the big papers in PLOS or Science or PNAS. There's a disconnect there between the consumerism of the data and the who's paying into that data and how the data is being credited at that level. So in some ways, I think the field has to wrestle with this fundamental problem of flashy results that are taken on the cheap in terms of not having this, not just recognition, but essentially paying into where this sort of, frankly, very altruistic component of this intellectual data is, which is, sort of at the curatorial level and all the things that entails, from the collection to the curation to everything else, to the, making the data available to the community. I mean, there are so many problems there with how we deal with this now. And I mean, I think there are many ways in which we could potentially, without even fundamentally changing the nature of the way we do science, there's ways in which, at the editorial level, for example, journals could be upweighting the way they could make it mandatory that people are doing things like feeding back into databases, for example, when they are, generating the types of data from specimens, for example, that you described in your talk, Rob, or other things where editors are taking into account, is there an original data contribution here, or are they essentially paying back into this community that they're essentially taking data from? As sort of something that becomes an extra bump into whether this thing actually, merits the standard of peer review or publication in a particular journal. I don't know, I think there's a lot of potential solutions here that we're not necessarily discussing as a field, but I think we should be.
Yeah, so Dan, one more comment on that thread. When we published this paper on Bourbon's role in eLife, we actually cited every single provider in the references cited. And admittedly, the way to do that now is to cite the DOI, right? You get the data, and this is now routine practice in my lab group. We never publish a paper where we're not citing a DOI back to the data that were used in that publication, so that basically GBIF can assemble the publication trail, so that the publishers get citation credit. And I think the thing that's going to have to change is, you're right. There's two sides of this, right? One is editors need to say, you need to show that you're a good citizen and not a data parasite, right? You're going to be buying into the community and working with them to basically make this a better ecosystem. I have a bunch of slides I didn't talk about that lay out my view on this matter, kind of because I think a lot about community and communities working together to curate and improve data. And until we actually view that as a collective enterprise, it's not about your citation impact, but about doing the work that benefits the big question, not the big questions. I totally agree, Dan, are the big macroevolutionary questions. So the big thing is, can we collectively work to solve problems that are going to save us from ourselves, right? And that's going to require us.
What do you think? What do you think the best way, in your sort of view, what do you think the best way is for that incentivization? I mean, as I see it, there's a big cultural disconnect now between, even if I look across the landscape of people I work with and collaborate with, there's a big disconnect in some ways between the people who are sort of plugged in the museum community who already think like that, and a lot of the others, most of the others who sort of haven't really assimilated that. So what is your sort of, if you were thinking about ways which we could break that down and sort of change that cultural landscape, which I think is really what has to happen here. I don't know what sort of ideas do you think might be effective at doing that?
Yeah, I kind of hope that maybe, like, I know that NSF has made more overtures in some respects around funding or to facilitate and make catalytic like team science approaches. And I think with this new Center for Open Environmental Data and Synthesis, a question will be whether that's sort of activity that will be community coalescent, can kind of spread social practices more broadly across a community in a way that actually takes root. And I think that the problem is that the center models have still been very much like, I do a meta-analysis, I publish a paper, and this goes into tree or something, right? And we need to stop perpetuating that model and have models where we are enabled to do team science, and the results are like this group, without any names associated with it, but this group produced this really neat community model that solves a problem. And I think, like, other disciplines have been much faster at getting there, but as I think a lot of our science becomes a lot more geared towards like trying to be more ultimately collaborative and inclusive, I think the push is there. But honestly, it's carrots and sticks. Like, the carrot will have to be the funding, the stick will have to be the journals and the editors kind of pushing against basically bad practices and demanding that, if you don't cite a DOI as a minimum when you publish a data paper, like, that's just not acceptable anymore. You just can't do it.
Big physics, be an example of a community that to some degree has done this, right? You look at the big Astro projects, the LHC project, other sorts of things. I'm not saying that there aren't problems there, but nonetheless, there is a more collective model that operates in terms of as a community with respect to the data projects.
Yeah, I mean, I think this has been like the conversation I've had for 25 years now about why we're not physics. And I think in part it's just because, there's always been place-based sort of individualistic, research programs that are perfectly reasonable and wonderful. I wouldn't want to give up on the idea that place-based research and PIs who have their slice of a puzzle aren't able to do that kind of work. But what I want to see is that instead of thinking about it truly as puzzle pieces that are basically scattered across this huge board of the globe, we actually are trying to fit the pieces together and make them connect, right? If we can do that, then maybe as those connections start to cohere, people will see that the laterality, this sort of ability to think about interdisciplinarity and team science, is actually going to be a better model and a more rigorous model for them to be able to do the kind of science they want to do. I don't do the science because I expect to get citations. I do it because it's super fun and it's interesting and awesome. And because I really believe in interdisciplinarity, I want to work with really interesting, broad people who do awesome things. And I mean, ultimately, I would much prefer that awesomeness than anything else. And so if we make it so that awesomeness is the if we make it so that awesomeness is the core of what we are talking about in terms of how people feel valued, I think that could start a process.
So not a great conversation, but to kind of switch topics here just briefly, because we're alluding a lot to the quality of data and the work involved behind the scenes. But as we kind of, and there's been several questions in the Q&A and a couple in the chat that revolved around data. And how do we, if we're going to the field, obviously we're collecting these animals, we're bringing them back into our collections, we're spending a lot of time and effort in making sure that data is as high quality as possible. But the original collection event that involves, whether it's taking measurements or taking tissues or parasites, whatever it may be, how do we optimize that to make sure that it's as useful as possible for users downstream? And I think it kind of gets to the extended specimen concepts that you referred to, Rob, and Dan, I know you mentioned that as well, but how do we make sure our collections are that really live beyond what was thought to be just a traditional systematics and taxonomy exercise, but a process that really engages a variety of stakeholders that are beyond the traditional, user group of a museum collection.
Hey, Dan, do you want to sort of take that one and start? I'll jump in afterwards.
Yeah, I mean, I think a lot about this, and I guess it comes down to, I don't have a good solution at the sort of museum end of things, but where I think about this is with the promise of essentially, I mean, we look at all these and actually this gets to something that we were talking about earlier about what, where in time a specimen is collected in terms of its value. And I actually think that right now the specimens we collect now, even in this disequilibrium world, in some ways are more valuable than ever before, because we can think really hard about these long-term uses and ways, the types of data that we can collect that weren't possible. And the most important data for me in some ways are what the type of data we're typically not really doing a good job of tracking, which when we think about extended specimen, I think that the word extended, going back to this idea of an extended phenotype, I like thinking about this idea of the extended collection situation. So these are all the properties of like the community, the ecology, the habitat, the abundance, and the sort of trapping method, and what that tells you in terms of reconstructing something about the population itself, what that community looks like. These are all these extended things that don't even sit cleanly within any typical sort of framework here in terms of data. How do we represent this stuff? How do we get people to record this stuff? Some of the stuff, people have done some of this stuff with like looking at, there's certain kinds of collection events that work very, very well with this. Like if you go and like fog a tree and a bunch of stuff comes out, well, we dump everything into, a single jar of ethanol, and we can literally reconstruct the species abundance distribution from that, fine. But many, many other types of collecting events don't actually fit that sort of clean model there. So how can we actually think about a forward vision of what collecting is that lets us maximize the amount of this sort of extended data that we're not going to get any other way for so many of these communities that are going to end up in a soybean field in the Amazon at some point in the future, or whatever, climate's going to change and vegetation community is going to change. And how can we actually make that available to the community in terms of some sort of we talk about ontologies and standardization conventions and so on, but how do we do this for these more complex ecological attributes? I mean, this is reflecting to some degree my bias and personal interest in the types of things I would like to do stuff with, but I feel that that's a huge, both an opportunity and a challenge, and I don't actually have a great solution. But yeah, I think it's out there, and I think it's something we should be thinking about, because this is sort of our we're going to the field in a lot of cases. We're spending resources on this, and this is a huge way of really maximizing long-term potential of some of these data.
Yeah, it's a great point, Dan. So I see from two sides. Like I think on one side, like we're going to generate a ton of really exciting data from the samples we already have, with their limitations about how reporting happened regarding the process, the event process that generated that sample. But at the same time, I mean, after having watched what is possible now with CT scanning, really the amazing strides we've made in using historical samples for genomics. There's a lot of the extended specimen that's going to be developed purely from sort of post accession, voucher specimen work, and that will be part of that huge network of data and knowledge and representations that sit around the physical material. And the physical material, to me, is the most important thing, right? It's at the center of all our graphs. But Dan's totally right. Like we need to do a better job of assembling the reporting at the inventory level about what happened when we did that inventory. And like it's fine. Like if your inventory is purely incidental, awesome. Like that's perfectly reasonable, and you can report Darwin Core level metadata. But if you're inventorying in a way that requires you to actually think about taxonomic scope or a sample method that is reportable, you should be able to report that as an event report with that relates to your specimens, that relates to the things that you collected in the field. And so I've been thinking about this forever. And finally, in 2018, we published a paper on this thing called the Humble Core, which was supposed to basically allow you to report the metadata about the inventory process, what happened during an inventory. But a lot of times we really do want to do planetary inventories. We're talking about planetary inventories as a really important piece of this puzzle. And when we wrote that paper, it was basically a concept paper. It had some terms and it had some, concepts behind it. But in the last few months, Paula Zermaglia, who was mentioned as a collaborator, and I and a bunch of others, John Machorik and a bunch of people from across infrastructures. This is ALA and GBIF and Map of Life. I've tried to really think hard about converting that into a usable extension for the Darwin Core, so that when you publish your records, if you have the inventory metadata, you can publish it in a way that's simple and clear. And I'll be honest with you guys, this is going to complicate publishing data. It's not going to be as easy as mapping fields and hitting the button to get Darwin Core. It's more complicated because our designs are nested and replicated in different ways. And it's going to be more of a pain in the butt to actually publish the data. That's okay. We walked down the road of learning how to publish the simple stuff. Now we need to walk down the road. I feel like this is all about journeys. My God. We're going to walk down the road to publish the hard stuff, and that's good. We'll get there if we're committed to the cause.
Anything to add, Dan?
Well, no, I would just say in some ways that's, you can imagine some of these conventions also evolving through time, and our ability to use data are always going to be contingent on whether we recorded things right to begin with, right? So actually, this is just, I guess, some comment for folks working in the field is to, I guess, I would suggest thinking hard about the way you record things like your effort, your sampling, the actual way in which you're doing your work in the field in a way that even if we have no way of actually thinking about how that's going to be served to the community now, that doesn't mean that in 10 years we won't. So, I guess that, my view is to just be as thorough as you can about every possible way you can, to the extent that you have the time and resources to do so, quantify the amount of effort you're putting into these sorts of things. And because that sooner or later we'll find ways of maybe making some of this comparable such that it can be used more generally.
Yeah, so Dan, I also would just say, I've been really fascinated to watch some of these thematic collections networks that are focused on species interactions and how much work we have to do to piece together the parasite that came from the vertebrate host. And we can do it, but the fact that we have to do this huge amount of work to link the specimen that went to one museum that is the host species, and then the other museum that has the parasite data, and to reconnect those dots at the specimen level. If we did that up front better, we wouldn't spend thousands. It's like you've tripled the work or quintupled the work to do it post hoc than to do it up front in the field, right? And so if we were able to be a little bit faster about kind of baking those practices into how we operate every day, it would be a lot easier for us to make kind of significant progress on basically specimen level host parasite relationships or any kind of interaction at all, even mutualistic like pollinators.
Yeah, great. Well, we're coming to a close here. There's actually, this is, I think, probably in terms of questions, a great set of questions to end on that are in the Q&A from graduate students that are looking at finding ways to leverage collections in their research, that are wanting to know how do we get students, high school age students, undergraduates, community college students interested in natural history work, and how do we train that next cohort of scientists that are going to come up and really be responsible for addressing some large scale questions that require this sort of the basic underpinnings of organismal biology to address climate change, urbanization. You mentioned pandemics. What do we need to do to train that next generation?
Wow, that's like the $1 million question. It's such a hard one. Dan, do you want to jump in here? I mean, I'm happy to do it, but maybe you want to start this one up.
I mean, I would agree. There's many levels to this question, right? I mean, from re-emphasizing these sort of ologies and but this isn't independent. I guess the first thing I'd say is that this isn't independent of the rest of our discussion here, because this leads to these ideas about even just training and just doing a better job of training students comprehensively to think about where their data come from and what goes into this and what goes into the data that might be, that they are using to, even if you're working with geographic distributional or taxonomic data, there's a cost to that data. It comes from something. And so just even instilling like an ethic into that, and where that comes, how we, in some ways, if you're in a museum community like Rob and I are, it's easier to work with this, because we are, the students who come through our program, they're working with the collections, they're sort of getting exposed to this over and over again. I have maybe fewer ideas about necessarily how we address that more generally. But it's something we talk about a lot here in terms of what kind of training we provide to our students, but how you address that with outside of a museum community is certainly more difficult.
Well said, Dan. I agree with the main point, and I think the thing that we're going to grapple with in museums in the next few decades and probably forever is really a huge set of issues around who can be in the museum and where our data originally came from, and that they're just very difficult issues about making sure that we actually live to the creed of being able to be really open and inclusive in a museum setting. And I'm not saying that museums, I think museums are the most awesome place to work in the whole world. Like I'm so amazed, literally amazed that I get to work at a museum. It is like, it blows my mind every day. But I think there are a lot of people who would feel the same way if given the opportunity to know how to get it in the door, and that the door existed. And I think that's just going to take a huge movement away from viewing it as business as usual in 2021 and in the next decade, around how we consider reaching out and pulling people into our worlds and telling them that they belong, that they're part of a community that can be very supportive and very nurturing to grow people who love natural history. I mean, look how many people use iNaturalist. Look how many. Tell me those people aren't interested in biodiversity. I mean, there are literally millions of users. How many of them are going to walk into a museum and be jazzed about all the stuff we have in our drawers if they knew how to do it? If they knew that there was an entrance, if their parents didn't tell them you're never going to make money in natural history, and if they were told that there's actually careers and really awesome work that can happen, that there are going to be data scientists in natural history museums in the next decades, that there are going to be people who will be able to do tons of really interesting things that are beyond just the science. They're going to be able to blend outreach and education. They're going to be people working on totally new modalities about how we look at the natural world and natural history, and getting those people in because they're the innovation drivers. They're the ones who are really critical for the next generation, right? That's the whole point of the symposium, is that there's this next generation coming. We're going to be ready to enable them to be successful in museums. We need to talk about this every day in every meeting about how to make that happen.
Just some really natural connections to students there, because you mentioned iNaturalist. I mean, just sort of re-establishing this valuation, this valuing of the doing basic natural history, of just going out and documenting what the heck is out there, right? And what things are doing in nature. And I mean, whether it's frankly even a museum or a naturalist, or ending up as like a herp note or whatever, like this process of getting basic fundamental natural history data and making it available in some form is something that many of us love to do, whether we're in a museum or not. It was inspiring to us as we started our own career, and I'm sure that there's a huge cohort of students out there who may not have been exposed to it, who would totally gravitate to that if they were exposed to that. So, and I think that that's a far broader sort of way of getting at this than just museums per se.
I think that's a good closing, and Rob, thank you for the nod towards the the innovation aspect of this entire thematic symposium. And I do want to thank Rob and Dan for having a really good discussion. It seems that most of the people were able to stick around to hear this, and I do want to remind everybody that every Friday from now until April 2nd, we'll be doing this with a different set of speakers from 1 to 3 p.m. Eastern. And we'll be getting into some of the early career folks that'll be joining us for the next couple of weeks, and then move in to our last keynote with Pam Soltis, one of Rob's colleagues at the University of Florida. So with that, thank you, Rob, and thank you everybody for joining.
Thanks, Cody. Thanks, everyone.
Thank you so much, guys. This was awesome.
Yeah, thank you, Rob. That was great. That was perfect discussion. So we'll, yeah, hopefully you can join us the next couple of weeks. So
Plan to.
all righty.
Yep. We'll see you guys.
Yep.
Bye-bye.
Thank you. Thanks, Dan. Linda, are we all set?